Hi, this is Shen. I'm a PhD student currently. I'm a former student with Claudia, and today we're gonna go over our newly published paper on the effect of prior and experiment design on the inference of precision matrix in a Gaussian chain graph model. So let's jump right in of what kind of question we're trying to answer. But first of all, like biologists aim to understand networks. These networks can be between genes, between microbes, between animals, between plants, etc. And the best way to know this network is actually by perturb them. For instance, you can kill this green microbe and try to see how other things response. Then here is this experiment design perspective. It is how to perturb this network. Like you can choose which microbes to perturb. And another question would be, what if you just don't know the effect of a certain treatment? For instance, you can throw some antibiotics into it, but you don't know which one killed. Well, if you are willing to assume some linearity, like for instance, we assume a linear model between any focal microbes and other microbe and perturbations, and we parameterize the direct effect of the perturbation or treatment and the interaction among these microbes, that will lead to a Gaussian chain graph model, which looks just like a multivariate Gaussian. You have this experiment, you have this treatment effect, but you also have interaction effect enter into this mean structure and also its covariance. And we will focus on learning omega, which can be interpreted as the interactions. However, under this model, that there are some internal confoundings, and this confounding can hurt experiment. Let's say this example, for instance, you have one treatment, and this treatment might benefit the first microbes but hurt the second. Or another alternative mechanism could be this treatment hurt one of the microbe, and these two microbes are competing that will look like this experiment are benefiting the second. And under this setting, like experiments might not be useful when these type of confounding exist. You can't really get away with this confounding by just changing experiment design because interactions are always there. Then what's the alternative? Actually, we show that prior might help. If you put some priors in the right place, that it can break these confoundings. For instance, in the first row, if you put the prior in the right place, you can see the posterior with and without experiment can make a very big difference. But if you put it on somewhat wrong places, that experiment won't really reduce your uncertainty. So what is the takeaway message in this entire paper? Is the overall question is what experiment to run after all these math. Our suggestion is first, we want to run the experiment that you know the most about the direct effect of it. And also you want that experiment to make most change in the marginals to the community. Thank you.